Before I start, let's announcement as usual. There is a biology colloquium this week, Thursday at noon. Your colleague, biology senior Jordan Lehman, will be talking about her experience with seed banks. Okay, here's the title: Seed Banks and Squatting, an epic experience in biodiversity and questioning all I've learned in biology. So come Thursday at noon and hear what Jordan Lehman has to say about that. This room, Thursday, noon, here. So we're transitioning today to talking about the endocrine system. So we're moving away from neuroscience and neurons and moving into endocrine physiology, anatomy and physiology, and hormones. And there's some real similarities. There are going to be some themes that you're going to have seen before, which is nice. But we will be branching away from neurons. And some of the cells that we're going to be talking about are considered neurosecretory cells. So they've got some neuronal morphology, physiology, and anatomy, and some endocrine properties. So here's an outline of what we're going to do today. So endocrine anatomy and physiology is the third and final section of this course. We will move from general endocrinology into specifics related to human reproduction, male and female reproductive anatomy and physiology. And that will be the end of the course. So today I'm just going to introduce some basic principles of endocrinology, talk about glands and their hormones, the different chemical types of hormones, how they are processed, which is important. Start to get into hormone receptors, and this is going to be a review and amplification of things that you've learned earlier in this course, as well as from cell physiology. And then we'll talk about, if there's time, we'll talk about forward and feedback signaling in the endocrine system. So hopefully you'll recall from cell phys that hormones are signaling molecules that are released into a vascular system. That's the definition of a hormone. Neurotransmitters are signaling hormones that are released into a synapse. And then there are other types of signaling molecules that are neither hormones nor neurotransmitters, like cytokines and other things that we're just not going to have time to talk about. So in the endocrine system, a cell from a gland that secretes a hormone. This is supposed to be a cartoon of a cell from some gland, and of course it's got all the things that cells have. It's got mitochondria, it's got a nucleus, it's got endoplasmic reticulum, it's got everything that the cell has, but this cartoon doesn't show it. It just shows a secretory vesicle releasing a hormone into a vascular system. Since anatomy and physiology, we're not going to be talking about plants, so I'm going to talk about blood, right? Exclusively. I'm not going to talk about phloem and xylem and all that other stuff. But plants have hormones too, just so you know. Hormones are released into a blood vessel. The hormones can travel a short distance and then be released from the blood vessel to its target, or it can travel throughout the entire body. In fact, it always travels throughout the entire body. It's just a question of where the target is. So for example, this gland might be the thyroid gland in the throat, and the cells that are responding to it are all over the body. Muscle cells, liver cells, heart cells, brain cells. The targets of thyroid hormone are very broad. They're everywhere. Or we could be talking about the release of another hormone. I'm just giving you an example. A hormone called ACTH from the pituitary gland, and it's released into the blood, and the ACTH goes everywhere, but its target, its primary target, is the adrenal cortex. So, adrenal, right above the kidneys. So it goes everywhere, but it really only, it has action primarily at one organ as opposed to everywhere. Goes everywhere, but its target is specific. So this cartoon is a little misleading in the sense that it looks like the hormone knows where to go, and of course it doesn't know where to go, it's just going everywhere, but it has either ubiquitous or specific target cells. Now what makes a target cell is that it expresses a receptor for the hormone. That's what defines the target. If the cell has a receptor for the hormone, because the hormone goes everywhere, the thing that gives it specificity is the expression of a receptor. And we'll be talking about those receptors pretty soon. And like I said at the very beginning of the class, some of the endocrine cells that we talk about are considered neurosecretory cells. They're not in the gland, they might be from the nervous system. But instead of releasing their signaling molecule at a synapse, in which case the signaling molecule would be called a neurotransmitter, sometimes the neurosecretory cell releases its signaling molecule and it travels into a bloodstream, in which case now we're talking about a hormone. So some cells that look like neurons are in fact neurosecretory cells because what they release is a hormone, not at a synapse, but into a blood flow. And then again, the blood goes everywhere, the hormone goes everywhere, but what is specific is the expression of a receptor on a specific class of target cells. They're neurosecretory. I guess yeah, they're neurons. They have axons.
So they're activated by a presynaptic neuron.
They are. They have dendrites. They are activated by a presynaptic neuron. They have axons. They release a signaling molecule. But in this case, it's not a synapse onto another neuron or a muscle cell. It's a release site onto a blood vessel. So here's sort of an overview of the types of secretion that we've been talking about. So this is what we were just looking at. You know a lot about the nervous system. You know that there's nerve cells and there's axons and there's targets. And in some cases the targets are other neurons, and in some cases the targets are heart or digestive tract or ciliary ganglion in the eye. So just remember, don't forget everything you've learned in the class so far about neurons. In cell phys, we talked about autocrine and paracrine, and we're going to need this knowledge, so I'm reintroducing it here very simply, because we're going to need this for some forms of endocrinology, especially reproductive endocrinology. So you'll remember that, I hope you'll remember, or let me just remind you, that autocrine signaling is when the cell that secretes the hormone, it's considered a hormone, but it doesn't go into a blood vessel, and it doesn't go everywhere. It stays local, and it might actually activate itself or its neighbors. So if the hormone secretes itself, it's itself. If the hormone secretes the cell that released it, it's considered autocrine. If the hormone diffusing locally activates neighboring cells, it's considered paracrine. Autocrine, paracrine, and usually they go together. And then exocrine is the secretion of things onto the skin, into the digestive tract, and we're not going to talk about that. Okay, let's do a clicker. This is to get a conversation started, to answer questions, and to ask you to remember things from cell phys. I'll read them as you're working. what is or are differences between signaling systems? The endocrine system requires a vascular system to transport the signaling molecule, but the others, autocrine, paracrine, nervous, don't. The endocrine and nervous system engage in long and short distance signaling, but the paracrine and autocrine systems engage only in short distance signaling. In general, the endocrine system has receptors with higher affinity for their hormones than nervous system receptors have for their neurotransmitters. And D, the cellular morphology of nerve cells is more complex than that of endocrine cells. So I'm going to ask you to show me your best answer. Okay. I'm fooling most of you most of the time because the correct answer is all of them. So let's go through them. So you all got it partially right, or at least if you put something up, you got it partially right. But I'm not sure anybody got it all right. The endocrine system requires a vascular. That's the definition of the endocrine system, and the others don't. So most of you chose A as the correct answer. The endocrine and nervous systems are both long distance and short distance. But paracrine and autocrine are only short distance because of the local diffusion of autocrine and paracrine signals. So that's a true statement. In general, the endocrine system has receptors with higher affinity. That's a true statement. And the reason that's a true statement is that the hormones go into the blood system, into the vascular, into the blood. And so they are diluted by an enormous factor. The concentration of hormones in blood is vanishingly small, picomolar sometimes, nanomolar. Very, very low concentrations of hormone in the blood. So their target cell receptors, in order to be able to bind hormone at such a low concentration at any significant level and do anything in response to that vanishingly small concentration of hormone, have to have high affinity. In the nervous system, the neurotransmitter is released into the synapse. The space is tiny. The release of just a few molecules of neurotransmitter into the synapse increases the concentration of neurotransmitter in the synapse, the tiny volume, is huge. It's millimolar, if you actually calculate it out. And so the concentration of neurotransmitter in a synapse is orders of magnitude higher than the concentration of hormone in the blood system, the blood. And one of the questions on your problem set, so let me comment. You have a problem set due on Sunday, and almost all of the answers can be gotten from this class and from Self-Phys. There's one that requires Thursday's class. So you can get to work on it now, but it's not due till Sunday night. One of the questions on the problem set asks you about this, so you now have the answer to one of the problem set questions. And it also asks you what's the structural basis for the high affinity that hormone receptors have for their hormone. What's the structural basis for that? And that's a Self-Phys level question, and I'll let you work on it, and you can ask me questions if you have questions. And the cellular morphology of nerve cells is more complex. Yes, this is true. Nerve cells have more elaborate dendrites, have generally longer axons. So at that level of morphology, endocrine cells, most endocrine cells don't have dendrites, don't have axons. They have release sites, but they don't have that. So they don't have the complicated cellular morphology of a nerve cell. And so this is a true statement. Generally, it's a generalization, but it's true. Okay, so now here's an overview of where we're going for the next couple of weeks. This is more of a reference slide than a teaching slide because obviously this is weeks worth of material on this one slide. We will be talking about certain glands. We will talk about their hormones. And then we will talk about their effects on the rest of the body. So this is a reference slide that you can refer back to. as we go through these, pretty much one at a time. I'm not going to teach from it now because that would be like trying to teach 3 weeks of material in the next 15 minutes. What would be the point? This is where we're going. I want to mention that there is endocrinology that we're not going to get into. You probably could have predicted that. It's more complicated than we're going to be able to do in 3 weeks. There are whole courses on endocrinology, whole medical professions of becoming an endocrinologist. So this is just an introduction. And part of the things that we're not going to do in this course is talk about the function of organs that you've heard of that secrete hormones. So the heart, in addition to pumping blood, secretes a peptide hormone. So it is, part of the heart is considered endocrine. And that's about all I'll have time to say about it. The gastrointestinal tract really has a lot of endocrine cells in it. That's part of its function. And it secretes a bunch of hormones. And they all have important functions. And we do talk about this in the other A&P course. So this will not be on any test. This is a reference slide for the things that we're not covering in this course. And in fact, we're not going to talk about another endocrinology. I will mention melatonin as a hormone that's secreted by the pineal gland of the brain. Some of you have been able to identify that in some of our brain sections. But we're not going to have time to talk about circadian rhythms. Our diurnal cycle, the fact that we tend to be awake during the day and asleep at night, mostly. So let's transition to a little bit of biochemistry, the biochemistry of the different types of hormones. And we're going to stay focused on 3 different types: the amine hormones, peptide hormones, and steroid hormones. And that's going to cover 90-something% of the chemistry of hormones. And some of these hormones you've already heard of. You've heard of epinephrine and norepinephrine and dopamine, because they're neurotransmitters too. So these 3 molecules, when released at a synapse, are considered neurotransmitters. And released into the blood flow, into the blood, they're considered hormones. You've heard of some of the peptide hormones, like insulin. And I'm going to add to that. And you've heard of steroid hormones, like testosterone. And maybe all the above. But I will hopefully add to your knowledge of the steroid hormones. So this is a slide, I think maybe a slide or something similar to what you've seen before when we were talking about catecholamines as neurotransmitters. These are amine hormones now, and these are the catecholamine amine hormones. And I am not going to ask you to learn the sequence of synthesis, but I am going to ask you to know that dopamine, norepinephrine, and epinephrine are some of the amine hormones. They're amines. There's the amine. They're catechol amines. Okay, so this is the structure of catecholamine hormones, and they all come from tyrosine. That's their source. That is key information. Tyrosine is metabolized by the cells that release catecholamines into the catecholamines: dopamine, norepinephrine, and epinephrine. This is a slide from England, which is why it has adrenaline. Noradrenaline. But the English, in America, in the United States, I don't know about Canada, in the United States, we call it norepinephrine. And in England, they call it noradrenaline. Why? I don't know. So these are the catecholamines derived from tyrosine. In addition, so now that's information you had before from the early part of this course. Now, new information. In addition to the catecholamines, the thyroid hormones, thyroxine, which is sometimes called T4, and triiodothyronine, which is almost always called T3, just because it's hard to say. This is the structure of the thyroid hormones. They are not catecholamines, but they are amines. Right? There's the amine. There's the amine. You can see the ring structure that suggests that they came from tyrosine as well, and they do. So the thyroid hormones, T3 and T4, come from tyrosine and are specifically secreted by the thyroid gland. They're synthesized and secreted by the thyroid gland. T3, why is this T3 and this T4? This has 3 iodine attachments to the ring structure, and this has 4. Iodine. That's a weird one, right? We don't talk about iodine very much. Iodine is an essential nutrient. If your body doesn't make it, you have to eat it, and your body needs it, requires it, for the production of thyroid hormone. If you don't have iodine in your diet, you can't make thyroid hormone, and you develop disease. These are not considered catecholamines because they do not have this structure. They have ring structures, but they don't have the catechol. Yes.
Is it just the two OHs that make
What do we call this position in chemistry? Meta orthopara. This is meta, right? Mm-hmm. The meta OH on the ring structure, on the benzene ring, makes this a catechol.
Okay, so we don't have both.
It has to have both. It has to have both to be a catecholamine. So you can see, so there's an OH that came from tyrosine. There's an O that got, what's the reaction? Condensation reaction, right? And there's the OH that came from tyrosine, but it's not a catecholamine. Like I said, just to introduce that there are hormones that are amines that are not from tyrosine, they're from tryptophan, and one of them is melatonin. So that's about all you need to know about the, no, that's not. That's where we're going to leave the biochemistry of the amine hormones for today. Now we're going to, this is now a review of key material from cell phys, because we're talking about proteins, peptide hormones that are small proteins that are secreted. And you'll remember that in cell phys we spent some time on the synthesis and trafficking and exocytosis of secreted proteins. So this is what we're talking about: the synthesis, processing, and secretion of peptide hormones, which are small proteins. So let's walk through this story. This is a story of, at the molecular level, of the synthesis and processing of a small protein that is destined to be secreted into the bloodstream by the cell that makes it. So it's a protein. So what is the name of this structure, this green blob? It's a ribosome. And what's the name of this yellow thing?
mRNA.
mRNA. So this is messenger RNA, right? We are reading the code for this protein from the messenger RNA. We are not seeing, well, tRNA, obviously tRNA is important for, what's the process? I'm just trying to review some of the language of self-is. What's the name of the process that's going on right here?
Translation.
Hearing the, is this replication? Is this transcription? Or is this translation? This is translation. Transcription is the making of messenger RNA from DNA, making of messenger RNA or other RNAs. Translation is the process of reading the code from an mRNA and making a protein. So this black strand is supposed to be the newborn protein being synthesized by this ribosome in the process of translation. Okay, is that clear? This is really important review of cell physiology. You need to know this. If for no other reason, then somebody's going to ask you on this on comps. It's going to happen. Some of you seniors probably already know that that's going to happen, or it has happened. Translation. Protein being synthesized. What is special about this red code, this red peptide, this red piece at the N-terminus? Remember, proteins are synthesized with the N-terminus first. So this is the amino acid that came out first, this red one. What is special about this 20 amino acid stretch of amino acids at the N-terminus of a protein that is due to be secreted? Anybody remember? Localization. Yeah, we can be more specific than that. Chemically, what is special about these 20 or so amino acids? They are going to bind to the receptor because what property do they have? It begins with an H. They are all hydrophobic. There is a hydrophobic stretch at the amino terminus of proteins that are destined to be secreted. So the code at the 5' end of the messenger RNA coded for a stretch of hydrophobic amino acids. That's the signal sequence. And that tells the cell, oh, this protein is not a cytoplasmic protein. This protein's got to get into a secretory vesicle eventually. Okay? So what does SRP stand for? Signal recognition particle or protein. It recognizes the signal. It has affinity for binding the signal. It has affinity for binding hydrophobic amino acids. What does it do when the signal recognition particle binds to the signal in a free ribosome? It tells it, stop translating. I'm not ready for you to go on yet. Because what needs to happen is this free ribosome has to interact with the secretory channel that's going to allow this protein to get into the lumen of the endoplasmic reticulum. So the SRP recognizes and binds to the signal, halts translation. The ribosome binds to this thing called the translocon. What membrane is this? Oh, there it is. It's the ER membrane. This is the membrane of the endoplasmic reticulum. So now we have a ribosome that is docked to the ER. There's a process to release. So there's a process that let's use the words. So there's a transmembrane protein in the ER that binds SRP when it is docked, when it is bound to a signal peptide. And when SRP receptor binds to SRP and hydrolyzes GTP, that allows the SRP receptor, that allows SRP to let go of the signal and allows translation to continue. So the SRP receptor and the translocon are essential for releasing the halt on translation and allowing the newborn protein to be translated through this channel into the lumen of the ER. I haven't added very much from what you learned at one point in self-is. So the protein, now translation can occur. The protein is being translated through the translocon into the lumen of the ER. This is called co-translational translocation. Co, same time as, co-translation. It's being translated and translocated into the ER at the same time. Okay, so this peptide is being secreted. And what is BIP? BIP is a chaperone, like HSP70, that stabilizes the protein. Yeah?
Is that considered part of the translocon?
BIP is a separate protein. It's a luminal protein that is a chaperone, and we're not going to talk about it very much. Remember that chaperones and like HSP70 and BIP are important for stabilizing and folding proteins. And that's important here. The protein has to be synthesized, stabilized, and folded. Is there anything else here that strikes you that is confusing? Because this is a key storytelling for peptide hormones. Yeah.
So the peptide hormone goes and attaches itself to the translocon, right?
Really, it's the ribosome that attaches itself to the translocon. And when this complex is formed, with the ribosome attached to the translocon and the SRP receptor attached to SRP, we have a functional complex that releases the halt and allows the co-translational translocation. The signal sequence also binds to the translocon, and you'll see why that's important in a minute. Okay, so what then happens? Well, we now have a protein that has been synthesized into the lumen of the ER. But there's been a step that is, so you need to write this on your slide. What happens here?
That comes off.
The signal sequence gets cut off by the signal peptidase. So you need to annotate your slide by reminding, and I gave you this one, didn't I? Yeah. That the signal peptide is cut from the newborn protein, the newborn hormone, and that releases it from the membrane, and so that the synthesis continues into the lumen of the ER. So we have cleavage by the signal peptidase of the signal sequence. And we have co-translational translocation of the peptide hormone into the lumen of the ER. Now we're continuing the story that you learned from self-is. The protein is folded. Yes?
Sorry, what was it called? What was it cleaved by?
The signal is cut from the protein by the signal peptidase, signal protease. Yeah. So now this used to be the N-terminus of the protein, and this is now the N-terminus of the protein because we cut it right here. Okay? So the protein is folded in the lumen of the ER. You'll remember that the lumen of the ER was important for forming disulfides. So if the protein has any cysteine-cysteine covalent bonds, they're formed in the ER. There can be additional cutting, additional proteolysis processing of the protein in the lumen of the ER or in the Golgi. So now we're starting to move into the Golgi. and there is a language that we need to speak. A hormone has a precursor called a prohormone. And in fact, this slide is incomplete, and you can add, did I give you this one too? Yeah. Add to it, the precursor to a prohormone is sometimes, not always, but sometimes, a pre-prohormone. Okay? What's the difference between a prohormone and a hormone? The prohormone is cut to make the hormone. I'm sorry?
You said that one last.
The difference between a hormone and a prohormone is that the prohormone is cut by a protease to make the hormone. The hormones get concentrated into secretory vesicles. The secretory vesicles are trafficked to their release site. And then we have the story that you are now very familiar with, calcium-stimulated exocytosis. This is just like neurotransmitter release. What is calcium binding to trigger exocytosis? Synaptotagmin. B snare, T snare, conformational change, fusion of vesicles, release of the hormone. That's secretion. So secretion in endocrine cells is calcium stimulated just the way secretion at the nerve terminal is, and the mechanisms are more or less the same. Yeah.
So I know that calcium.
Same thing here. Same thing here. We're going to talk about depolarization. So endocrine cells fire action potentials. They do everything that neurons do: receive signals, process signals, depolarize, fire action potentials, have voltage-gated calcium channels, have calcium-stimulated release of a neurotransmitter, in this case called a hormone. They just don't have axons. Most. Some of them do. That one we would call a neurosecretory cell. A neurosecretory cell as opposed to just a secretory cell. So endocrine cells are sort of honorary neurons in a way. They just don't have the complicated morphology of neurons. Of course, an endocrinologist would say neurons are just fancy endocrine cells, whereas a neurobiologist would say endocrine cells are just simple neurons. Okay, so let's give an example of this processing of a peptide hormone. And this is the most important example of processing of a peptide hormone. In fact, you'll need this to solve some problems, maybe on the problem set or maybe on a quiz or a homework. So this is an example, but it is the most important example, and I will use the specifics of this example because what we are going to get to are some important hormones like ACTH, which is one of the ones I mentioned at the beginning of class, and endogenous opioid hormones. So these are important hormones that we're getting to, and here's the story for the production of these hormones. These are all peptide hormones. So we're still in the peptide hormone story. What is synthesized by the ribosome is a protein called POMC. Which is not a hormone. It's a pre-pro hormone. What is synthesized by the endocrine cell, in this case, it's a specific cell in the pituitary, we'll get to that, is POMC. So here's the carboxy terminus, and here's the N terminus. The signal was over here. POMC. Pro-opiomelanocortin. Pro-opiomelanocortin. Pro, because it's a precursor. Opio, melanocortin. Do you think there was a pre-pro-opio-melanocortin? Yes, there was. Where was the signal on pre-pro-opio-melanocortin? It's not shown here. Was it out here, or was it over here?
On the left.
It was on the right, or on my right, on your left, on the N-terminus. There was a signal sequence, and when the signal was cut off, the pre was cut off. Now we're left with pro-opio-melanocortin. And it's a protein. It's 260 amino acids. I don't even know if it has any disulfide bonds. It has specific sites where proteases cut it into bits. Protease, protein cutters, protease. So there are proteases that cut here and release the hormone ACTH. There are other proteases that cut right here and release this whole protein. Well, let's not worry about that. Let's talk about this one. There are proteases that recognize the specifics of the structure and cut here and release this little peptide, which is called enkephalin, metenkephalin. There's another one that is cut here and here, or actually it doesn't cut here, it just cuts here. So if this is cut without this being cut, we release a different peptide called beta-endorphin. You've heard of these words, endorphins and enkephalins are the endogenous opioids. So depending on how POMC is cut, it can release ACTH, or metenkephalin, or beta-endorphin, or alpha melanocyte stimulating hormone. Different hormones can be produced by POMC.
So just to be clear, those parts that are cut out are not the hormones, they're just part of the alternate.
No, the part that is cut out is the hormone. This is not splicing. This is just proteolysis. This piece that my fingers are bracketing is ACTH. The rest of it is either proteolyzed to make either, so if we cut here and here, we can release both ACTH and metenkephalin.
Interesting. Okay.
Yes?
Can you cut like everything all at once? Or?
Yeah, well, if you cut everything all at once, then so if we cut everything all at once, we would never make beta-endorphin because we would only make metenkephalin. And if we cut everything all at once, we would never make ACTH because we would always make alpha melanocyte stimulating hormone. So that doesn't happen. Yes?
Are there specific used for these
Sure. So there is a specific one that makes ACTH. And there's a different one that probably only cuts here to make melanocyte stimulating hormone. And we can regulate them independently. So we can say, we don't want to make it metenkephalin. We want to only make ACTH. And so the hormones that cut out here, the proteases that might cut out here could be turned off, and the protease that cuts here and here could be turned on, and we would make ACTH only, or primarily. Yeah.
For example, what happens to the rest of it?
Oh, recycles.
Okay.
Yeah. Uh-huh.
So is it more about the proteases that do the signaling as to which component they're cut out, or is it like the signaling?
There must be complicated mechanisms for the control of the proteases that I actually don't know much about. Because if they were all uncontrolled, just like I said, if they were all uncontrolled, we would always make alpha melanocyte stimulating hormone and never make ACTH. So there's got to be control to limit the cutting of here so that we can make ACTH, for example. But the main point is that POMC is the precursor of a number of important hormones. And by precursoring, I mean that we make the precursor, POMC, do specific proteolysis to release peptide hormones. Would I ever put up this map and ask you, without the labels, and say, which amino acids are cut to make ACTH? No, I would not do that. Would I ask you to know that met and enkephalin and ACTH share a precursor? Yes, I would expect you to know that. Would I expect you to know that POMC is the precursor to opiomelanocortins? Yes, I would, because that's embedded in the name. But I would not ask you to know where the specific locations on POMC are the specific hormones. It's not worth it. Okay, let's do some steroid hormones and then we'll take a break. So we've done amines, we've done peptides, and now we're going to do steroids. And then we'll be done with the chemistry, biochemistry of hormones. All of the steroid hormones are derived from cholesterol. That's their defining feature. So if I show you a structure of a steroid hormone, if I just show you a structure, you should be able to identify it as an amine, no. As a peptide, no. As a steroid, yes. It's got the ring structure. So you can see, here's this 4-ring structure. Cholesterol has the fatty acid tail, the steroid hormones don't. They are amphipathic, but they're not as lipid soluble as cholesterol because they don't have this fatty tail. What's the word hydrophobic tail. But they're partially, they have some hydrophobic nature and some hydrophilic nature. They're amphipathic. Will I ask you to be able to identify aldosterone as opposed to testosterone? No. I will not ask you to be able to identify the different steroid hormones from their structure. But these are some, not all, of the important steroid hormones. Progesterone, female reproductive endocrinology. Aldosterone, kidney function. Cortisol, a steroid hormone that is important for all the cells in your body. In fact, of all the hormones that we talk about in this next section of the class, cortisol is the one hormone you can't live without. Cortisol is absolutely required for life. If you have a defect in the production of cortisol, you don't live very long. That's not true for a defect in the production of testosterone. If you're a male and you don't make testosterone, you've got some issues, you've got some problems, but you can still survive. If you're a female and you don't make estradiol, you have some problems, especially reproductive problems, but you can still survive. If you are either a male or a female and you can't make cortisol, you don't live very long. Cortisol is required by all the cells in your body to live. Aldosterone. If you don't make aldosterone, your kidneys are going to be dysfunctional. You could have some renal problems, but I think you could still live. Okay? Bottom line, steroid hormones, all from cholesterol. Cortisol is required for life. Where do they come from? Well, they all come from cholesterol. And as predicted, they have a complicated map of synthesis. So let's look at the important ones. Where do they come from? Am I going to ask you to be able to regenerate this slide? Of course not. Am I going to ask you to be able to label this slide? You know, if I put in boxes like I do sometimes, you know, and ask you to fill in the blanks? No, I'm not going to ask you to know that. But I am going to ask you to know, to be able to interpret a map like this. In fact, I'll do that on the problem set. So let's take a break now, and we'll come back so that you can refresh a little bit. and then we'll come back and talk about the key points of this map of steroid hormone synthesis. Steroid hormone synthesis, where I will try to emphasize the things that you do need to know and things that you don't. Let me make one comment about the key concepts list that was here and was posted on Moodle. And that is, on the bottom of page 1 and on the top of page 2, you can see that it's blank. And this is so this key concept list is acting at least a little bit like a worksheet. So as we go through this, you can go back, maybe at the end of class each day, go back to the key concepts list and say, okay, we talked about aldosterone. I don't remember us talking about the release site. We didn't talk about it. But we did talk about the target, kidney. So you could already start to fill in some of the key concepts. The target of aldosterone is the kidney. Cortisol, we talked about it, and I mentioned the release site, maybe too quickly for you to catch. Target, everywhere. And so use this as a worksheet, and at the end of the section before the exam, I will make sure that all your blanks are filled in. Okay? All right, synthesis of steroid hormones. We could spend a whole semester on this, but we're not. Cholesterol is the precursor of all of them. That is a key testable fact. That is a key fact to know. So this is the precursor. This is the product of an enzymatic reaction, and this is the enzyme that catalyzes this reaction. So P450-SCC stands for side chain cleavage, P450 side chain cleavage. I will not ask you to know the names of any of the enzymes. But they all are enzymatic reactions. They all are catalyzed by specific or sometimes non-specific enzymes. So the P450 side chain cleavage enzyme cuts cholesterol into pregnenolone. Pregnenolone is not one of the ones that you need to know. And then the HSD. What is HSD? I didn't bring my decoder key, so I don't even remember what HSD stands for. Something decarboxylase. You know, it's the name of some enzyme. Yeah.
So are these all continuously cut? Like, do they all get smaller?
Do they all get smaller? That's a good question. Let me see. So the enzymes that were hydroxylase would be cutting off a hydroxyl. Side chain cleavage would be cutting off a side chain. Hydroxylase, aldosterone synthase. I'm not sure.
Okay.
Probably not.
Okay.
But they're not getting much bigger or smaller. They're all pretty much, they're changing around that core of the cholesterol ring structure. There could be some that are adding on, like a carboxylase, that would add on a carboxyl group instead of a decarboxylase, which would be taking off a carboxyl. So now we have a branch. Pregnenolone is acted on by the 3-beta-HSD enzyme to make progesterone. That one is a hormone to know. And it's also acted on by an enzyme called the P450 17-alpha hydroxylase. So two different enzymes, and depending on which enzyme is working on the, what do we call this? This is now a substrate. Depending on the enzyme that is acting on the substrate, we get two different products. Do you need to know that pregnenolone is the precursor to all of the steroid hormones that we're talking about? No. Cholesterol is. Pregnenolone is also, but no cholesterol. Do you need to know that progesterone and 17-alpha hydroxyl pregnenolone are the two products of action on pregnenolone? No. But the point is, cholesterol is processed into progesterone, and then the precursor to progesterone and progesterone are processed both this way by a series of enzymes and this way by a series of enzymes. Okay? Different enzymes, different products in the map. If we just follow this pathway, we've got a P450C21 enzyme and then a P450 aldosterone synthase, P450 aldosterone synthase, P450 aldosterone synthase, the same enzyme working on different substrates, make aldosterone. What do you need to know? Aldosterone is a product of cholesterol. How many times do I have to say it? But it's not the only product of cholesterol. So here, the P450 and 17-alpha hydroxylase is acting on either pregnenolone, progesterone, or deoxycorticosterone. Right? The same enzyme working on different precursors or different substrates, just like the same enzyme working on different substrates and making different products. Once we get over to this stream, the 3-beta, so this is the same enzyme working on two different substrates. This enzyme here and the P450C21, the same one here. P450C11, that's a new one, P450C11, making cortisol, the one hormone you must have in order to survive. So if you have a defect in the P450C11 enzyme that renders it non-functional, you don't live very long. And I'll ask you in the problem set to be thinking about what would happen if you had a defect in the P450 17-alpha hydroxylase, or what would happen if you had a defect in the P450 C21 enzyme. You have to look at this map to answer those questions. The P450 17-alpha hydroxylase enzyme also, remember we did it here, now it also acts on these substrates to make the precursor to the reproductive hormones. What is different? Well, so the dotted line suggests here are things that happen in the adrenal gland, making aldosterone and cortisol. And here are things that happen, well, these have to happen in the adrenal gland, and these have to happen in the gonads, testes and ovaries, in order to make the male and female reproductive hormones. So I could ask you to use this knowledge and say, what would happen if we have a defect in the P450 17-alpha hydroxylase? Would testosterone levels be higher or lower or normal? Would aldosterone levels be higher or lower or normal if we have a defect in P450 17-alpha hydroxylase? So let me ask you that question. I don't even remember. This might be one of the questions on the problem set, but it might not be. If there was a defect in the P450 17-alpha hydroxylase, what would happen to concentrations of testosterone? They would go down. What would happen to concentrations of cortisol? They would go down as well, because we need that hormone, that enzyme, to get over into this stream. This person probably isn't going to live very long, but let's pretend that there's just enough P450 17-alpha hydroxylase to allow survival. What's going to happen to the concentration of aldosterone? Higher, lower, or normal?
Increase.
Increase. Why would it increase? Instead of going this way, we're going to shunt it this way and make more of this pathway. That is a key concept. If we inhibit one pathway, by mass action, we're increasing the concentration of the substrate, the precursor, and allows the other pathway to go more. That's a key concept.
But there's no controlling of protein expression that inhibits the over.
Oh, I'm sure there is.
So can we make the assumption that a certain.
Let's not get too complicated. So let's just use this map and not say, well, there must be an enzyme that breaks down aldosterone and is going to keep the concentration of aldosterone normal. That's probably true, but let's not do that. So you have to use the idea that if you inhibit a hormone, if you inhibit an enzyme, things upstream of that enzyme can back up and increase. So you'll use this map on the problem set. Okay, let's do a clicker. What is or are differences between hormones? The steroid hormones are all hydrophobic or amphipathic, but amine hormones are all relatively hydrophilic. Peptide hormones generally have a higher molecular mass than the other hormones. Peptide and amine hormones are required to sustain human life, but steroid hormones are not. Peptide and amine hormones require proteins for their biosynthesis, but steroids don't. Which of these are true differences? Really true statements. Okay, I think we need to go through this. The best answers are A and B. Steroid hormones are all hydrophobic or amphipathic. Let's go back to their structures. There it is. Steroid hormones are all, this is a pretty hydrophobic one. This is a pretty amphipathic one. So they're all amphipathic or hydrophobic. Amine hormones are water soluble. So they're hydrophilic. They've got lots of polarity. So that's a true statement. So A was true. Peptide hormones have a higher molecular mass than the other hormones. Yeah, I mean, the steroid hormone and the amine hormone, we're looking at atoms, and you can count them on, a couple of hands. Whereas with peptides, we're talking about 10, 12 or so amino acids. Each of those amino acids is 100 Daltons or so. So a 10 amino acid steroid hormone has a molecular weight of 1,000. The steroid hormones and the amine hormones have molecular weights of 100 or something. Right? So peptides are bigger. They have a higher molecular mass. Peptide and amine hormones are required to sustain life, but steroid hormones are not. Cortisol is a steroid hormone. That renders this statement untrue. Peptide and amine hormones require proteins for their biosynthesis, but steroids don't. That's a protein. It's an enzyme. It's required to make cortisol. It's required to make aldosterone. So steroid hormones, they don't have peptides as precursors, but they certainly have proteins that are required for their synthesis as the enzymes in the pathway. Okay, so that was what that question was asking you to realize. So A and B are correct, and C and D are false. Okay, so now, big transition. We've talked about the hormones. Now we're going to talk about their receptors. Big transition. Hormone receptors. And this is all review. All of it is review, either from the beginning of this course or from Selfies. So I'm not going to do it, but you should. You should do it, because this I could test you again. Some of the hormones bind to a receptor that is coupled to GS. It's a GPCR coupled to GS. Some of the hormones bind to receptors that are coupled to GI. You know, or should know, and should refresh your memory of, the GS story and the GI story. I'm not going to do it again. But you should, if you haven't got it deeply ingrained in your memory. Here is a list of the hormones that interact with GS-coupled GPCRs. And here's a list of the hormones that bind to and activate GI-coupled GPCR. Now, do you think I want you to memorize this list and this list?
Heck yeah.
No, absolutely not. I do not want you to memorize this list and this list. Please don't memorize this list and this list. But if I tell you somatostatin interacts with a GI-coupled GPCR, tell me the story of how somatostatin works on the cells that express its receptor, you should be able to tell me the GI story. And some of this is on the problem set. So, or I could give you this list and say, when norepinephrine and epinephrine bind to beta adrenergic receptors, what happens to the concentration? What's the second messenger, and what happens to it? Does it go up or go down? You should be able to answer that question. Cyclocane P goes up. That's the second messenger in the GS story. That sort of thing. So for example, I think on the problem set, I ask you about the activation of D1, what happens when dopamine binds to the D1 class of dopamine receptors. And I think I ask you about, okay, do not memorize the list, but remember, memorize, learn the story of the receptor and its activation of a second messenger and protein kinase A or other protein kinases. And you don't have to know all of this either. The point, what is the point here? Protein kinase A phosphorylates and activates many substrates that have physiological importance. And GI leads to the inhibition of protein kinase A, which changes the activation of many proteins. That's the main point here. Do I want you to memorize all of these effects? No. Too many. What do you suppose the next slide is? GQ. Here's the GQ story. Here are the hormones that activate GQ-coupled GPCRs. Remember the GQ story? Don't memorize the list of hormones. Protein kinase C. This one actually this is probably the best GQ slide I've got. There's the protein kinase C story, right? Activation by both calcium and diacylglycerol. Protein kinase C is activated by diacylglycerol and calcium. But here's another wing of the calcium story. Calcium binds to calmodulin. Calmodulin-dependent protein kinase is another kinase that phosphorylates many things. Okay? And here are the hormones that activate these pathways. Okay, so refresh your memory of GPCRs. What's going on here? This is going back to cell phys. What type of receptor is it? Oh, it's up on the screen. These are receptor tyrosine kinases. So now I'm asking you, for the first time in this course, to review the mechanisms of receptor tyrosine kinase, because we need to understand them to understand three important hormones. We're going to talk about insulin, you've heard of that one, growth hormone, you probably heard of that one, and prolactin. We're going to talk about the effect of prolactin when we get to reproductive endocrinology. These three hormones interact with receptor tyrosine kinases. So let's review the story. Binding, diffusion and binding, and maybe you can annotate your slide here. Diffusion and binding. Dimerization of receptor subunits. This is review, but it's been a while. Dimerization of receptor subunits. Cross-phosphorylation of receptor subunits. The receptors are kinases. Receptor, tyrosine kinase. They phosphorylate each other when they are dimerized. On what amino acid? Tyrosine. The dimerized, cross-phosphorylated receptor recruits a number, so it's not shown here, but remember there was that recruitment of adapter proteins. So annotate your slides here saying binding of adapter proteins and activation of kinase cascades. Everybody got all the labels that you need to annotate your slide?
What was the last
It's
one?
already-
What was the last one?
The last part was activation of a kinase cascade, a series of kinase activations. And they're all tyrosine kinases, actually. Other questions about the receptor tyrosine kinase story? Will I ask you to know that insulin, growth hormone, and prolactin are activating this pathway?
Yes.
Yeah, why not? It's only three. So you can memorize these three, and everything else is not in this story. So if I say progesterone, you should know we're not going into the receptor tyrosine kinase story. In fact, if we talk about progesterone, we're going into the third and final class of receptors, the intracellular receptor. So here's the hormone in the blood. That's what this H is, the hormone in the blood. It might or might not be carried by a protein in the blood. But there are carrier proteins that bind and help get that hormone distributed throughout the whole body, sometimes but not always. What is going on with this arrow, this pair of arrows here? What is this process? Diffusion. This is, remember, these are hydrophobic or amphipathic. These hormones can diffuse through membrane because they are hydrophobic or amphipathic. They diffuse, passive diffusion, no transporter needed, passive diffusion through membranes. What's happening here? What is this equilibrium? Come on, give me the word. It's B. Binding. Binding of the hormone to the receptor. What is not shown here begins with a C, two C's. Conformational change of the receptor hormone complex. What's going on here? Transport of the receptor hormone complex to what? Where are we? The nucleus. That's where the DNA is. And what is this hormone receptor complex doing to DNA? It's acting as a transcription factor, either a positive or negative transcription factor, activating the transcription and then the translation of response proteins.
It is the transcription.
The hormone receptor complex is a transcription factor. The hormone is not a transcription factor. The hormone receptor complex is a transcription factor. And here are the hormones that follow this pathway. Basically, all the steroid hormones and the thyroid hormones. So you've memorized two things, steroid hormones and thyroid hormones, and you know you're not in a GPCR, you know you're in an intracellular receptor story. So there are things to memorize. You have to memorize these three to know that you're in the receptor tyrosine kinase story. You have to memorize these two, thyroid and steroid hormones, to know that you're in the intracellular receptor story. And everything else is a GPCR, and I don't want you to memorize which GPCR. That's the thing. Okay, so we'll start on Thursday with feed forward and feedback signaling. That's where we'll start on Thursday. I'll see some of you in lab this afternoon.